My name is Arya Sadafi and I'm coming from Helmholtz Center in Munich. I'm also affiliated with Chair of Computer-Aided Medical Procedures at TUM. I'm presenting our work about attention-based multiple instance learning for classification of blood cell disorders. Blood disorders are a good example of natural selection process. For example, in sickle cell disease, people with just one mutated gene are highly protected against malaria, while people inheriting two genes from their parents suffer from severe anemia. And in Africa, where malaria is one of the biggest killers, up to 40% of people are believed to carry one of the sickle genes. So we decided to develop a method to analyze blood samples because blood samples are easy to obtain, a blood test is cheaper than a genetic test, and manual analysis of blood samples is time consuming and requires expertise which might not be available in those affected areas. Several approaches exist for classification of red blood cell shapes. In this publication, authors are suggesting a deep neural network for classification of red blood cells for diagnosis of sickle cell disease, with a dataset of 8 patients and around 7,000 oxygenated and deoxygenated cells. In another work, a method is designed for outlier-tolerant classification. Using a deep regression network, two shapes of a sleeper and croissant are expected, and they find the transition point between the two shapes in images of red blood cells in flow. Finally, in this work, an active contour model is used to segment the cells, and some basic shape analysis descriptors classify the cells based on their circularity and elongation. However, all of these approaches only refer to one type of disorder, or they do not take into account the proportion of misshaped cells in the diagnosis. The diagnosis of blood cell disorders requires analysis of several images, and not all of the cells or regions of interest are relevant for the diagnosis. And enough number of relevant cells need to be found in order to decide about a patient's disorder. Automatic classification of the samples is more accurate and speeds up the process, and it should mimic the clinician by looking at different cells all together for a diagnosis. So in our work for classification, we decided to use a multiple instance learning approach, which takes into account every cell in the samples and works with weak bag labels, so the expert does not have to annotate all of the cells in the dataset. Clinical adaptation of any method requires explainability, so we decided to use attention-based pooling in the multiple instance learning solution in order to provide better insight about the network's decision to the clinicians. Also, an auxiliary single instance classification branch helps us to converge better. The samples that we use for this study are coming from Relevance Consortium and are microscopied in the University of Zurich. We used bright field microscopy and the cells are not attached to the surface. There are 5 different classes in the dataset: healthy, hereditary spherocytosis, sickle cell disease, piezo1 mutation, and thalassemia. We have a total of 60 patients and over 80,000 cells. For pre-processing, we decided to use an RCNN architecture to detect the cells. Rich features extracted after region of interest aligning are used as an input to our model. We trained this part on a previously annotated segmentation dataset, and we are using it for single-cell detection. Here is our proposed architecture that is using the features that are extracted by the RCNN and embeds them into vectors of 500 numbers. These vectors are then passed through some other convolutional layers that are helping us to calculate the attention scores for every instance. After having the attention score per instance, we do a matrix multiplication and calculate the bag feature vector. And with the bag feature vector, we try to classify the bag. The auxiliary single instance classification branch is also introduced here, trying to classify each of the instance vectors into one of the defined classes. This branch receives a higher importance at the beginning of the training, and as we move on, its importance gets less. For this purpose, we are defining a dynamic class, giving more importance to the single instance classification loss at the beginning, and as we move on with the training, multiple instance learning loss gets a bigger coefficient. Here you can see images from 3 different samples belonging to the patients of thalassemia, sickle cell disease, and spherocytosis. After training, model not only is able to correctly classify these samples, but also it tells us which cells did it use most for this decision. Here, after testing some samples with the model, we are listing the cells that receive the highest attention during the classification. We can clearly see the different patterns that are picked up by the model in a self-supervised manner for different classes. In spherocytosis, the cells become spherical, or in thalassemia, the membrane is not formed correctly. Sickle cell disease also makes the cells look like a sickle. And all of these patterns are correctly picked up by the model, so it makes us sure that the model knows where to look and what to find for a classification. Quantitative results are also supporting this. We designed an ablation study with 4 different models and did threefold cross-validation on 5 different runs. We are reporting the accuracy, F1 score, and area under curve for single instance classification, multiple instance learning with max pooling, multiple instance learning with max pooling and auxiliary single instance classification, as well as our proposed method. And we could achieve an area under curve of 0.96, which is pretty good for our task. However, our method has some limitations as well. For example, it is depending on the backbone network for feature extraction, which might not be available always for every dataset. And in case it is available, there is no guarantee that the features that are extracted by this network is useful for the multiple instance learning task. And multiple instance learning requires lots of data which might not be always available. Among the future works that we assume for this project, I can list measurement of the uncertainty of the model, finding out about the relevance of uncertainty score to attention values, or including the backbone network in the model, and doing knowledge distillation for initialization of this backbone network for an end-to-end training. And currently we are using a modified version of this approach for classification of a dataset of acute myeloid leukemia patients. And as you see, different patterns are picked up from different classes of the dataset. So I will conclude here and thank you for your attention.